Sources of bioelectric potential. As you know, our body is made up of different kinds of cells. Cells form tissues and tissues form organs. For various functions, body generate their own monitoring signals which contain some useful information. Bioelectricity refers to electrical potentials and currents occurring within or produced by living organisms. These are ionic voltages produced as a result of certain electrochemical activity by certain special types of cells. The existence of electric potentials at rest and during excitation is proved by the Italian scientist Galvani in 1700s. The electric potentials generated in human tissues are widely used for diagnostic purposes such as diagnosis of nerve, muscles, cardiac, brain, visual and auditory functions. The cell is surrounded by a semi-permeable membrane which separates the intracellular fluid from extracellular fluid that contains charged atoms like sodium, potassium and chloride. Potassium ions and negatively charged anions are present at higher concentrations inside the cell than outside. Sodium ions and chloride ions are present at higher concentrations outside the cell. The concentration differences for sodium and potassium are due to the action of a membrane active transport system which constantly pumps sodium out of the cell and potassium into it. It is this concentration differences for sodium and potassium, especially potassium, create the membrane potentials. All the bioelectric potentials are basically generated due to the diffusion of these ions across the membrane either during resting or at any moment of excitation. The cell membrane permits some substances to pass through the membrane while blocks other. The resting membrane potential is determined mainly by two factors: the differences in ion concentration of the intercellular fluid and extracellular fluids, and the relative permeability of the plasma membrane to different ion species. The cell membrane contains protein channels or leak channels that allow ions to diffuse across the membrane. In addition to leak channels, which are responsible for resting potentials, cells have voltage-gated channels for sodium and potassium, which allow the movement of a large number of sodium and potassium respectively. Ion channels that mainly allow potassium to pass from higher to lower concentration are called potassium channels, and ion channels that mainly allow potassium ions to pass are called sodium channels. Potassium leak channels open in resting neurons, others are closed and only open in response to a signal. Every time potassium ion leaves the cell's interior loses a positive charge and positive charge builds up on the outside of the cell membrane. Therefore, inside of the cell becomes negative relative to outside, setting up a difference in electric potentials across the membrane. Therefore, all cells under resting conditions have an electrical potential difference across the plasma membrane such that the inside of the cell is negatively charged with respect to the outside. This potential is a resting membrane potential. Because there is a potential difference across the cell membrane, the membrane is said to be polarized. The value of the resting membrane potential varies from cell to cell but usually ranges between -60 to -90 millivolt. In non-excitable cells such as epithelial cells and adipose cells, the resting membrane potentials does not change appreciably over time. In excitable cells such as neurons, muscle cells and some endocrine cells, upon stimulation of the cell, the membrane potential can change dramatically for short periods of time. Therefore, in excitable cells, the membrane potential is not always at the resting potential. Now, when a cell is excited, the semi-permeable membrane changes its characteristic properties. It allows the entry of sodium ions inside the cell. This neutralizes some of the negative charge inside the cell and makes the membrane voltage less negative. When the voltage approaches a threshold value called the gradient potential, the gated channels for sodium opens and a huge amount of sodium will be coming inside. then the voltage is going to increase as more and more sodium rush inside. Thus, when the cell is excited or stimulated, the outer side of the cell membrane becomes momentarily negative with respect to the interior. This process is called depolarization and the cell potential changes to approximately plus 20 millivolt. When the voltage reach around 20 to 30 millivolt, the sodium gated channels will get inactivated and the potassium gated channels are going to open. Then we have a rush of potassium outside of neuron and repolarization takes place. This process of changing from action potential or depolarized state to the resting state is called as repolarization. Passage of sodium ions from the outside of the cell outside to the inside of the cell is blocked. By an active process called sodium pump, sodium ions are quickly transported to the outside of the cell and the cell becomes polarized and assumes its resting potential. The rapid rise and fall in voltage of membrane potential across a cellular membrane is called action potential. The cell action potential shows a finite rise and fall time. A cell may be caused to depolarize and then repolarize by subjecting the cell membrane to an ionic current. However, unless a stimulus above a certain minimum value is applied, the cell will not be depolarized and no action potential is generated. This value is known as the stimulus threshold. After a cell is stimulated, a finite period of time is required for the cell to return to its pre-stimulus state. This is because the energy associated with the action potential is developed from metabolic processes within the cell which take time for completion. This period is known as refractory period. In neurons, action potential play a central role in cell-to-cell communication by providing for the propagation of signals along the neuron's axon towards end of an axon and then to the adjacent cells. In other types of cells, their main function is to activate intracellular processes. In muscle cells, for example, an action potential is a first step in the chain of events leading to contraction. In beta cells of pancreas, they provide release of insulin. Though minus 70 millivolt is considered as a nominal value of resting potential, the actual potential is derived from the Nernst equation. Em is equal to 2.303 RT by ZF log to the base 10 of AO divided by AI, where Em is a resting potential, R is a universal gas constant, T is the thermodynamics temperature in Kelvin, Z is the number of moles of electrons transferred between membranes, F is the Faraday's constant, AO is the concentration of ions outside the membrane, and AI is the concentration of ions inside the membrane. Substituting the values of the constants, we get Em is equal to plus or minus 61 log AO by AI. The figure shows the electrical activity associated with one contraction in a muscle. The wave of excitation while propagating in the muscle causes its contraction. This phenomenon is found with the skeletal muscles, the heart muscles, and the smooth muscles. Every contraction of a muscle results in the production of an electric voltage. These voltages are called action potentials because they are generated by the action of the muscles. After complete contraction, repolarization takes place, resulting in the relaxation of the muscle and its returning to the original state. The bioelectric signals, which are produced by the coordinated activity of large group of cells, which are often recorded for clinical purposes. In this type of synchronized excitation of many cells, the charges tend to migrate through the body fluids towards the still unexcited cell areas. Such charge migration constitutes an electric current and hence set up potential differences between various portions of the body, including its outer surface. Such potential differences can be picked up by placing conducting plates or electrodes at any 2 points on the surface of the body and measured with the help of a sensitive instrument. These potentials are highly significant for diagnosis and therapy.